So welcome everyone to the Abbvie Bioinformatics Challenge at HackIllinois 2021. I'm pleased to virtually meet you folks, I think under different circumstances. I've been to hackathons both virtually and in person. They have each their kind of pros and cons, but I think we look forward to kind of engaging with you folks in this format and seeing what you come up with. And just as a bit of background, I'll get to the background later. But I'm just really happy to see so many folks joining, and I appreciate the interest ahead of time. So this is a rough agenda. We'll go over kind of who we are. We're going to introduce some of the topics that you'll be working on if you choose to do the challenge, both at a high scale and like a little bit in depth, and then we'll go over the rough strokes what the challenge looks like. So to kind of get started and contextualize who we are, we've put together some information on Abbvie. So we're a biopharmaceutical company, an innovation-driven, patient-focused, specialty biopharmaceutical company that our main focus is, as we call it, to have a remarkable impact on people's lives. And this is, we do this through kind of modern approaches, but we have a long history where we've spun out of a company that's over 130 years old, serving patients in the healthcare industry. We started as a new focused company in 2013, but regardless, every day millions of patients benefit from our therapies and our drugs. In terms of kind of the areas we focus, they're in immunology, oncology, virology, neuroscience, women's health, eye care, aesthetics, and other specialties. And over time, these grow, but we're in most of the countries in the world in terms of having patients. We have over 30 brands of marketed drugs, and these encompass around 60 or more conditions. And so we have a large kind of R&D organization that researches new science, researches new therapeutics, and my organization is there to support and advance both IT as well as kind of analytics, data science to further that goal. And so we're really, the organization that I'm a part of, that I lead, we are here to help that mission, to bring new therapeutic options to patients across the world. And so what we're going to be talking about and what my team does is a lot of kind of computer science, computing, data related topics, but I think most folks remember and keep in heart what the company overall is trying to do. I'm sorry. And we're also going to introduce here the Abbvie Innovation Center. So this is kind of both a facility and an organization that started in 2014 in Champaign, Illinois. So it's part of our it is part of the BTS, which is Business Technology Solutions. That's the company's IT organization, and part of the R&D IT organization, Information Research. So this is our innovation hub and our academic engagement hub. There's different activities that go on, like the one we're doing right now, but I think the kind of marquee kind of effort that happens out of the AIC is the 60 to 70 students that work at Abbvie with Abbvie scientists. The majority of them are from the University of Illinois at Urbana-Champaign, but these are kind of all over the US, and maybe Jerome can correct me. I think we have had international as well. And there's different pillars of kind of what the students engage with in terms of and grow in terms of working and interacting with the AIC. But the bulk of the time is with these project experiences, as we call them, where students are working with scientists or information technology team members to bring something forward, to kind of create a new solution, or do analyses. And I found that these are kind of mutually beneficial for everyone involved. They're kind of meaty real world projects that kind of let students apply what they learn in their formal education as they're going through school. And so they get to see the applications of what they learn, and as well as kind of have something to point to, that kind of a completed substantial task. But in addition to those projects, the AIC also supports the students, and from my perspective, the organizations with onboarding, offboarding, professional development, and social engagement. And so, it's a really great program. I think we're kind of through this challenge here. This is like a mini example of like some of the type of work that happens, but the work is only one part of what the AIC does for the students. Do you want to add anything, Jerome, before I move on? I'm going to assume no. We'll allow some question and answer at the end. So we're going to get right into a kind of 30,000-foot overview of biology and how that intersects with programming, computer science, data analysis. There's different terms. One of them that's used often is bioinformatics. But we don't really have time to go over a full introduction to this field. But the way I look at it, so my background is actually in bioinformatics. I studied both biology and then statistics and programming. I capped it with bioinformatics. So the way I look at it, and I'm kind of expressing it here, what motivates kind of using code and engineering, programming and software engineering in biology is that it's kind of twofold. One of them, biology and biotechnology, is producing vast amounts of data. It's kind of infeasible for an individual to analyze quote unquote by hand, or even with kind of semi-automated methodologies. A lot of science traditionally is done with Excel, or the analysis of is done with Excel, or even more manually with calculators. That we have technologies now that will not scale to. It's not feasible, and so tools are needed, and the data is in some cases so large that the tools aren't inherently obvious. So it requires kind of a thorough knowledge of algorithms, data structures, to kind of come up with solutions. So that's one of them. Yeah. So kind of they're really kind of the tools needed require more sophistication in computer science techniques, we'll call it, to just get the work done. But there's kind of another one, and this is what has interested me in going into bioinformatics after kind of first approaching my education through a biology focus and lens. And that's that there's this part called molecular biology, which we'll get into. But a lot of what goes on there, what you learn in the science and what happens in living cells and viruses and organisms, it can be expressed in terms of information encoding, storage, and transfer. And so really what is happening in real life and how we, on a scientific basis, describe it is expressible in terms of communications theory and computer science methods. And so the application of these have both helped describe some of the processes that go on, but also infer some of the biological processes that go on. And so there's interesting interplay that's gone on in the literature around molecular biology and bioinformatics, and how that's informed, been moved, or has moved kind of computing topics. And so that there's a lot more I could go into in depth there, but I'll just say that there's some inherent kind of computer science occurring in real life, so to speak. And so that's what's interested me, and that's what's kind of enabled a lot of work in the biology field to kind of move forward through the application of computing science methodologies. So on a very simple basis, the human genome that we have, every living cell in your body has around 3 to 3.5 gigabytes of strings. They're just strings, they're letters, all from 4 letters. And it describes everyone. It describes you. Every single one of your cells has that 3 gigabytes. And your parents shaped what that was, but it's all there. And so there's an incredible amount of, as I said, that information processing and storage that occurs to make you and to make every other living organism. Just so that we can kind of describe the motivations of the applications that have come up that we'll touch upon later. It's worth kind of having the basic description of kind of what these molecules do. And I think folks might be familiar with DNA. That is the genetic code that higher organisms encode, and we do. So as I said, that 3.5 gigabytes is stored as DNA in your living cells, and it undergoes a transformation into another molecule called the RNA that's quite similar, that allows kind of control, and it allows information processing to occur. So the DNA is kind of like the cold storage, so to speak, although we know now that it's not quite as cold. There's activity that occurs there. But for the purposes of just describing kind of the information processing, we'll say that DNA is like the primary storage. It goes into the RNA, where the cells, depending on the conditions, process that, and then that then encodes, and it's kind of communication theory stuff. There's a encoding that happens there from RNA into these things called proteins, which are made up of 20 odd amino acids that then make up every part of your cells and every part of your body. Those are the things that are active. Those are the things that kind of you see in your skin, or that kind of produce your bone. Those are also, in terms of the pharmaceutical industry, those are the things that we kind of make drugs against. And in some cases, we make drugs using proteins. And so this is called the central dogma of molecular biology. And we know now there are many, there are exceptions that are starting to come up, but essentially there's this end product protein that all of which is encoded in DNA, and there's information processing that happens to get from the DNA to the protein through this RNA step. And so there's essentially, these strings are being copied and manipulated every minute of every day in all of your cells to kind of make you. If we so kind of having this framework, if we move on, the whole field of bioinformatics had either its second or third wave in the 90s with what was called the Human Genome Project. And so this was kind of current for folks of my age, but for some of you on the call, I think it might be a historical artifact, but nevertheless, this was an effort through large academic institutions partnering with industry as well, in a concerted effort to sequence, and sequences kind of elucidate the letters that make up the human genome. And so it was a large marquee project of science in the 90s, and the goal of which was to kind of figure out what those letters were for people, and the idea being that with that we would have a bigger lens to see what is going on in the cell, in organisms like ourselves. And along the way, we sequenced other organisms as well. If you look through here, there's different landmark. The mouse genome was achieved earlier before the human one. Nevertheless, this kind of doing this was a big effort. So in 1990, it was not otherwise possible in 10 years to sequence the human genome. Normally, it would take, as we said, there's about 3 billion letters. In the past, the throughput was kind of a couple thousand of those letters in a kind of 3 to 4 month period. And so if you were to just scale that, it would almost never end. And so new laboratory techniques were developed to cover the entire genome. And along with those laboratory techniques, new programming techniques, new bioinformatics tools came out. that led to what was called shotgun sequencing, where copies of the genetic information were made, chopped up, and then read using some newly developed methodologies. And this created kind of this soup of little fragments that it was then up to programming to pull together to get the final sequence. And so this enabled what would have been a multi-decade effort to get compressed and kind of be done in that 10-year time span. And actually, a lot of the work kind of was backloaded after the methodologies were developed and proved. It accelerated and got finished to create that first human genome. If we kind of fast forward to today, the technology has been refined and scaled up to what's now called next generation sequencing, which there's some experimental nuance between what that original human genome shotgun approach was and what is done today, but it's essentially the same, where you cover the space of an entire genome, of that entire sequence, in little pieces, kind of we'll say stochastically. It's not quite random, but you can call it random. And then you find the letters of each one of those using machines. And then it becomes a large computational problem to assemble, as it's called, those tiny fragments, which are strings. At that point, they're strings in a computer file. The assembly is kind of a non-trivial computational task. In the example we're going to use here, what we're talking about is there's the so-called reference genome. So this might be like the quote-unquote reference human genome that was established in that human genome project, the initial version of which at least. But so that's essentially like a string search problem. It's just on a large scale. And it's on a large scale where you have to support variations of which. It's not an exact string search problem. And so you can imagine so that there's tens of thousands to millions of these little fragments that then you have to match, which are usually anywhere from 150 to 400 letters long to that reference, which is 3.5 billion long. And so that's the problem. That's the problem that had to be solved. It continues to have to be solved in this part of bioinformatics. If we move on, so this is kind of more description of why. Another word for this is alignment. And so why is alignment hard? As we said, it's kind of, there's the 3-odd billion base pairs, as they're called, or letters. And it just takes a long time to kind of do that string search. And as I mentioned, they're not always exact matches. So there can be differences, which are called substitutions, but there's also things called insertions and deletions, where an individual organism might have extra letters in a given area compared to the reference. And so if you just kind of just do math, the exponents just get really big really quickly. And so it's not a problem that can be so-called brute force. Conceptually, the problem is simple. You take every substring of the reference and every substring of each query that you have, each one of those little fragments. You come up with a scoring function, you compare them and find the best. You find the best spot that it goes into. But that, again, it's not feasible. These numbers would just go up and up and up, and it would take more time than the universe has been, than the universe has existed to do it with a naive methodology. So how, what has been done in the field to kind of solve for this? There's multiple methods, but we'll just go over some of them that are here. They're all kind of making use of approximations or heuristics to quickly find what is the most likely match versus go over all of the possibilities and then rank them. So there's kind of, in broad categories, there's kind of different searching methodologies. So a lot of the data structures you might or might not have studied and search methodologies, you can think about them here. So kind of in the introductory methods, you can go over and think about which ones have different time complexity for the given inputs with the different search algorithms. But if in data structures, you might have gone over search tries and other search suffix arrays and whatnot. So these are all used and evaluated to kind of come up with what is the quickest way to do that string search. Another one would be kind of compression or compression/encoding. So the reference genome that you're after is very large, but it's very large. It's made up of only 4 to 5 letters. And so there's active research and applications of the research that has gone on to using different encodings and compression methodologies to fit that into a smaller area and to allow quicker algorithms to go over. And so there's a common one that's used in communications theory, and it's applied in some algorithms that do the sequence alignment and assembly task with the Burroughs-Wheeler transform. So this kind of represents the sequences in a different way from the inherent statistics in there, and it accelerates the process. And so you can kind of look into that if any of this piques your interest. So that has applications outside of biology. It was brought into biology from kind of general communications research. And the final one would be dynamic programming. And so we're going to kind of go into the explanation of that a little bit more right now. So if we take a look, the classic example of dynamic programming would be kind of top-down versus bottom-up solutions of recursive problems. So if we take a step back, and I described the core problem of aligning 2 sequences is, you have the rough naive algorithm is you compare every substring of given sequences with a scoring function, so that there's a natural recursive kind of formulation of that problem. And so if you were to naively implement it, as we were talking about, it would take a long time. But more specifically here with kind of the classic example of recursive function, the Fibonacci sequence, the kind of bottom-up solution would be kind of recursively doing it step by step. So you kind of keep calling it, and this tree, this call tree, as I think of it, just gets, the call stack gets bigger and bigger. And so the successive steps refer and reform the calculation of the one before. And so this is kind of the normal way you would describe that in terms of the Fibonacci, calculating the Fibonacci through a recursive function. But so what we're showing here would be kind of that, again, that computational tree, the call stack, as I think of it, of if you were going to do it with the direct recursive way, top down, but so from the request down. But if you were to do it kind of bottom up and keep track of the results, so every time you needed the Fibonacci of 1, you already had the answer, and every time you had the Fibonacci of 2, you had the answer. So you go through and figure out what you need and store the results effectively. This is another way to think of it. Then the problem becomes more feasible. And you can see here in this example code that it's making use of a table to store those results, and it enters the recursion kind of differently than the direct problem. Another concept here would be kind of memoization, and then the dynamic programming then is kind of using memoization and a different approach into the problem space to quickly solve it. And so, yeah, so this description shows here that the kind of intermediate results are stored in that table so they can be looked up, and the kind of execution of the whole problem is done a little differently to make use of that. This is kind of a rough example of dynamic programming. I think I would assume that you folks might have had a larger introduction to it in kind of an algorithms class. If not, there's resources out there that show what this is and its application to sequence alignment specifically. Nevertheless, so it's kind of taking the naive problem and attacking it piecemeal and storing the results to kind of get something that's computationally feasible. If we, this is kind of another example that this is used in, it's kind of the traveling salesman problem, or the kind of the optimal path problem in graph theory. Given some constraints, and so you have to kind of know the domain, you can split the task up into sub problems and then kind of find the optimum of those sub problems, combine them, and then hopefully, if your constraints were correct, have the optimum of the entire problem. So in this example, the shortest path between A to F, you can divide it up. If there's certain constraints, that you'll never travel backwards, then you can look, between E and F, for example, or some other substructure. And so this is kind of a graph theory explanation of the same concept, and it's used in kind of designing the different sequence alignment algorithms. And so for the interest of time and allowing questions, we will be distributing the slides in this talk, but I'm going to talk over mainly the overview of this example. But it was, again, as I said, with the example of finding the best structure, the sequence alignment, it's similar to the problem of coming up with the shortest path through a graph traversal problem. And so you kind of, given valid constraints, you chunk up the sequence alignment problem into smaller pieces, solve for those, and then solve for the bigger problem. And so this is an example of an actual algorithm called BLAST that kind of combines these different techniques as well as the encoding technique to solve the problem of taking a short sequence and finding where it matches in a large sequence, and by short, a couple hundred letters long, to a long one, the long one being the size of a whole organism, like in our case, so 3 billion. So this is using the kind of divide and conquer aspects, efficient search algorithms, and efficient encoding algorithms to do that. And so this is kind of a graphical representation of the algorithm, but you take the query, so that's a short one, and you take its substrings and you take it through a scoring function that determines kind of a high entropy or a high likelihood of search encoding, for lack of a better term, and of the different subsequences. And of those most, as I like to think of it, the most interesting subsequence, you take that, and that algorithm was then ahead of time computed for the subsequences of the reference genome. And so you're able to, and there's efficient, quick search data structures that can take a small set of letters and find them in a large set. And so what this does is it quickly finds the locations in that 3 billion where you would have a possible, well, a match, or a very, in this case, an exact match of this little interest area. And that lets you skip whole swaths of sequence. So this is kind of as I was talking about, dividing the problem into smaller ones with constraints. The constraint here is that you've identified a sequence that you think won't change. And so there's theory behind and math that goes on this scoring, what was determined that wouldn't change. Nevertheless, that is the kind of assumption that lets you constrain the problem and simplify it. And so in step 2 of this algorithm, you would then find of this given query where this little piece all across where it matches. And then you go through and do the slower but more flexible search methodology of the generalized sequence alignment between those pairs. And usually pairs are found. But nevertheless, this is an example of that optimization, breaking the problem down, using encoding and search efficient search data structures and algorithms to do it. And so this BLAST thing happens really quickly, and happens at a kind of genome scale. And so we're gonna, the hint, if you do the challenge, you'll be kind of making use of this through a web service here. But independent of a hint, you can kind of go in and look at the NIH has a kind of bioinformatics arm called the NCBI, and they have public servers, and the software is public, so you can kind of download it and run it yourself. But there are public servers that you can do this on, and the nucleotides, so nucleotide kind of DNA or RNA, you can kind of go into there and paste a given sequence of interest, kind of set their parameters. You can optionally say, maybe human, or maybe you don't want it human, but it will go through and check that query sequence, as it's called in bioinformatics, against the references, whether all organisms or some organisms. And kind of what you end up with then is kind of a list of where your query sequence hit, to what reference and where, and some quality metrics. So if we look here, the example that was put in, it has, of what matched, it was 100% the same. Identity kind of is a measure of sameness of sequence. And so anyway, you can go through here and look, and then if you were to look at one, it actually shows where the query, how it matches to different areas of the genome and where. So in this case, how to interpret this is that, in whatever this line is, you can kind of hover and see it. The query matched here and here to a given target, to a given real gene somewhere else, and it had a difference right here. But this part was unmatched. There was a gap, so to speak, or in the case of the query extra sequence, that looks like a gap with respect to the reference one. So you can play around with it and do this in real time. So this, again, what would normally be an intractable computational problem or near intractable computational problem runs in near real time. You can paste a sequence of different lengths, and within minutes, it's compared against sequences of different organisms that each might have conceivably billions and billions of sequences in their genome. So, and this bench scientists use things like this all the time to determine either experimental results or to design experiments if they're kind of designing, if they're making new DNA or new proteins. And so we've listed some examples of kind of the in-depth of the the computing and data algorithm, the algorithms and data structures behind the short read alignment problem and kind of alignment in general. So we're gonna get to the challenge now. So this was, the prior section was not meant to be exhaustive. It was to kind of show what is being done. I will say that as someone that practiced bioinformatics, a lot of time you need to know what that was, but a lot of the work day to day, you're using tools that exist out there. You need to know how they work, but you don't necessarily need to write them yourself, because the work ends up being stitching them together in the right order, and then creating automation around it, kind of doing the programming around it to make it repeatable. And then there's like a secondary analysis, so to speak. So after you might get, in the case of something like this, let's say that you had scientists that every week they were going to get a table like this, and so you might come up with an algorithm then to kind of do the picking like they would. And so a lot of kind of day-to-day bioinformatics is using the base tools, stitching them together, and then doing analysis on top of them. And so we've kind of recreated a similar task here with the challenge. So the scenario is, imagine 2046, a space rover has returned from its mission to an outer planet carrying plant and soil samples. And so hidden in here is, you can assume that these were sequenced. So if DNA was found in them and the DNA was sequenced, they've come to you to apply bioinformatics tools and a bit of programming to identify anything of interest in there. And we're telling you that there's a potential virus in the sample. And so if you find it, then you can locate kind of a cure or a code for the vaccine, and potentially also visualize results. So this is a kind of a imaginary scenario, but there have been people that do bioinformatics through either the kind of on the data analysis side or the operations side. All the time they're presented with, hey, we have these sequences, or we have these new results that are coming from a new type of experiment. We need you to help us make sense of it. And so a lot of times that makes sense of it is kind of a collaborative effort. So the person that's applying the tools and the programming on top of the results might not know all of the biology, and so they work closely with them. But nevertheless, this is kind of a fanciful example of a real workflow that happens in laboratories all the time now. So to the kind of specifics of the challenge, we've provided sequencing results in bioinformatics tools in a Docker container. So we'll get to that in a couple of slides, but you have conceptually everything you need, the data and like those bioinformatic tools that implement kind of the stuff we talked about, the different search tool methodologies, divide and conquering. It's all in there. You don't have to re-implement that, but you just have to kind of stitch them more together in the right process. So we have kind of 4 tasks here. The first one is to just describe the virus. And so you'll see what the files look like. There's kind of little sequences that we talked about. You'll need to use a tool, and the tool, there are many tools that can do it, but the hint here is that there's this one, SOAP de novo 2, that can assemble it. And assembling means taking those little fragments, lining them up, and finding what the whole one looks like. And then when you get that, you can actually kind of describe the length of it, maybe where it came from, what genes are in there. And so that's like a half of a hint. And so in this slide deck, there's examples of kind of how you might figure out where it came from if you kind of look at that slide deck. And then there's a hidden message in the viral genome. And so what we didn't get into in depth here, we can maybe put another hint, is that as I showed in that central dogma, there's a mapping and encoding between DNA to RNA to protein. And we didn't kind of go over that. It's not very complicated, but with the right Google search, you can kind of get at what that translation table is effectively. And if you do that, then you would be able to maybe look at it and see if there's that hidden message. That's an exercise left up to the challenge acceptor to kind of figure out what this translation is or isn't, and how do you do that via programming. It's not too hard. You just have to kind of find that right kind of coding matrix or translation table. These are different words you might want to search for. So if you've done these 2, then you can kind of propose a cure in the unfortunate case of that virus, if it were to attack Earth. And kind of a fourth challenge is the virus. There's standard ways that scientists can kind of visualize the genetic code of organisms or genes. And so this is kind of a programming and data visualization task. So you're free to come up with some of your own, like you'll have some data and some numbers, and you can think about how you might want to convey that to other people. Another hint, you can kind of search for this term plasmid map and see what comes up, and see how you might approach producing one of those. And so these are the main tasks. And I opened up the chat. It looks like people are asking about the slides. We'll make sure that they go up on the Discord. This is a super practical one. So there's a container for this hackathon, as we said. We felt this was easier to package up the data and those bioinformatic tools in one. This is the main command here to kind of run it. So I think there's really a step 0. If you haven't, step 0 of the challenge, which is if you haven't used Docker before, step 0 will be kind of familiarizing yourself with the use of Docker and how to get into this specific container. And so this will start it up and start an SSH server in there that you can then log into. So you need to kind of choose your own password here. The left side stays constant. The right side, you put the password you want for your password. The container will start up, and then you can interact with the container either through SSH or through other Docker mainline invocations. You can change the port. This is the port that would be on your computer that maps into the container. On the actual system, there's Miniconda kind of for kind of scientific Python environments. There's a separate node installation for those that prefer to write in JavaScript. There's an R environment with common packages. So I think a lot of bioinformatics classically had people wrote in Perl, then Python and R. So R and Python are the two big languages in bioinformatics, and you might or might not be able to find ready-made visualizations in here as a hint. And then finally, just a kind of more generic Python 3.8 environment with BioPython. So they'll all be in there, kind of dig around. The actual raw data will be in the container and /data. So take a look at what's in there as well. And then finally, how do you log into this? So you can either, if you haven't changed the 2022 port forwarding, you can, on your computer, on like the main laptop or desktop, you can log in via SSH. Or you can write this command after, you have to find the container ID of the container you created. You can find that out from Docker PS. This command line will directly give you a shell into the container to interact with it. So this is kind of the bare minimum crash course into how to interact with Docker. But once you're in there, you can treat it as like a regular Linux computer. You effectively have root in it. So if you have, some other programming language that you like using, if you want to solve this problem in Rust and kind of get the Rust toolchain up and running, you're more than welcome to do so, and you will be able to in that Docker container environment. Finally, if you get past step 0 and solve any of the tasks, we'll be collecting the results via SurveyMonkey. So please submit the results via this link. And otherwise, good luck. I hope you'll get a feel for what it is like to be a practicing bioinformatician. So thank you. And I guess I'll monitor the chat for any questions that come up, but I'm going to open it up to the Abbvie team if they have any comments they want to share with the students, with the participants, or comment on anything I missed. Thank you.
Just in terms of some of the logistics, the SurveyMonkey will be open till 5 p.m. Central Time. I heard that we have some folks on Pacific as well. So it'll be open till 5 p.m. We are giving prize for responses in the quickest time, as well as a prize for the best visualization. We are also accepting resumes for a bioinformatics incubator internship that we are organizing this summer. And we're collecting those resumes through SurveyMonkey as well. Looks like there's some chatter in the chat room around the code. Chloe just posted an updated code in there. So that's it in terms of the logistics. Pass it back on to Suku's team for anything else to add with the bioinformatics or questions from students for us at Abbvie.
I think the rest of the team on our side I haven't heard or seen any comments, so we're good on our side. Yes, so I think someone will share the presentation on Discord. Is that right?
Yeah, it's already on Discord under workshop. So if you go to the workshop, I'm new to Discord, so whatever the hashtag or channel is for this, for workshops and talks, I've posted the slide deck there, so you have all the links and all the information that Suku just presented.
Can we just work on designing the visualization? I think if you want to do that, that's fine. You're going to have to find someone else that's found data to visualize, or you make it up. I'll say it like that. As it stands, the visualization, what you would visualize, there isn't the inputs to that aren't there yet. The data we've provided is kind of raw. So there's multiple steps that happen. So that raw data gets combined using some tools in step 1, and then step 2 and 3 would be kind of looking at that using other tools to glean something out of it. And at that point, then you would have what would go into a visualization. That being said, my background was originally in science. I got used to seeing what it looked like to other scientists. That doesn't mean that's not the only way to visualize it. There might be something else visualizable in that raw data that we otherwise didn't think of. So visualization one is kind of open-ended on purpose. So I don't think you can work on the you can work just on the visualization, but I would be more than happy to be proved wrong.
From a logistics standpoint, we have required the first 3 responses before
you have
the visualization. So the one unless you find somebody else to work with the team for the first 3 questions.
A great question. Let me see if I missed any. I think it was the login questions, the codes, slides, and that final one, the visualization. So.
Rishin and Chloe, I think we'll turn it back to you if there are no other questions from the participants, from everyone else.